Well, good morning, folks. Welcome. Good morning to everybody. Hope you're all feeling well this morning. We're just kind of, in the early weeks of the school year or the academic year. So maybe you still have the energy to get up this morning. But thanks to everyone for joining us today. This is 3301, the genetics of mental health. So I guess I don't have a lot to say in the preamble department. I can just kind of just jump right into it, talking today about variation and polymorphisms or polymorphism, and then a little bit about behavioral genetics as well. So let's just jump right into it. So this is the topic for today's class, and we're kind of getting into we're beginning to gradually transition into stuff that's more, about what you're here to learn about, which is the way that genetics and epigenetics influence the brain and behavior. We're still, I think, still sort of in this semi-revisional preliminary stage, but hopefully working closer and closer toward getting into the good stuff. My aim here is not to give you an all-encompassing study or view of the different sources of variation and polymorphism in the genome. It's certainly not to give you an all-encompassing view of the different types of mutations. Some of that would be things you might better learn in biology or biochemistry or genetics, sort of typical biology genetics course. Others of them, I think, are things that we can cover as we go, so that if there's a specific mutation relevant to a particular disease that we're covering, that would be the right time to look at that, not right now. So I just want to give kind of a general review, and the same applies to behavioral genetics. I just want to give a kind of an overall survey of the territory, and we'll get into more of the finer points on a topic by topic basis. So we have previously praised DNA. Let's just see here. So I have a question coming from Simon. Are these slides posted on Brightspace? Not yet. So my system is to post the slides after the lectures. And I explained this the last time we met. The reason for this is I usually find mistakes in my slides as I present them. And so I'll, correct myself as I'm talking. But I don't like having to go back and revise the slides on Brightspace or CU Learn or whatever, because then people might have the wrong slides, and then the exam material could be messed up if somebody's studying off the wrong slides and so forth. So I've always found it to be a better policy to post them afterward. For now, just take notes from what you see. And yes, and good morning to everybody too. Thanks for your participation. Okay, so we had previously praised DNA for its fidelity. The kind of the big signature trademark of DNA really is that it copies itself, quite reliably from generation to generation. This is what enables. This is essentially what enables the stability of life, right? I mean, the first living creature to replicate itself wouldn't have survived very long if its genetics hadn't been able to be maintained somewhat faithfully. However, it's not completely faithful. There has to be some room for variation, otherwise evolution couldn't take place, and variation between individuals also couldn't take place. We would all just be clones of each other. So meiosis, as we talked about, allows us to create novel combinations of genetic variants. You get a little bit from mom and you get a little bit from dad, and you kind of end up being a totally unique person. But where do those variants come from in the first place? Well, typically you would call changes to the DNA sequence mutations. And so we're all genetically distinct, of course, except for monozygotic twins. And so in a certain sense, we're all sort of mutants. There's no prototypical human that defines, like, this is what the human genome is supposed to be. Anybody who doesn't have this exact sequence is somehow a mutant. It's discussed, I think, more in a kind of a relational or probabilistic way. You say that, like, a given genotype is unusual among a certain group of people. That would be something of a mutation. If it's novel, like if you have a new a new variant that neither of your parents had, that would certainly be classified as a mutation. But typically when we're talking about this, we use the term mutant for rare sequence variations. So things that have a less than about a 1% frequency, allele frequency in your given population, or de novo variants that are unique to an individual. Like I say, de novo, a de novo variant is just one that you didn't inherit from either of your parents. It means it mutated at some point, either in your parents' gametes or in you during your embryonic development. What's more relevant, I think, to us, though, is the normal spectrum of variation, which you would call polymorphism. And this is Greek for many form. Many is poly, and morph means form, so it just is many form. I tend to favor the term polymorphism because it doesn't imply abnormality. If you were to call every variation a mutation, then you could easily, you could look at a population that's very different from your own, from a different part of the world, and say, look at how many mutations they have. They're so different from us. That's not the right way to look at it. The correct, interpretation is that there's a varied and large gene pool that exists in the world, and humans come in, genomes come in many forms. So it doesn't, it's a bit more of a favorable term. So in the genomics age, a great deal of the genetic variation that exists in our planet, frankly, but in our populations, have been characterized and studied. So this is, I think this is sort of worth emphasizing, because it's been known for many, many decades that humans differ from each other genetically. And even before the human genome was first sequenced in the early 2000s, it was possible to look at specific areas using very laborious, time-consuming techniques and show that this is an area where, a bunch of humans differ from each other. Typically, those were actually like tandem repeats and like fragment length polymorphisms and things that don't even really have a lot to do with the actual function of genes, just other bits of DNA that were in there. But anyway, the variation was always assumed, but unless you can really go in and look at everybody's DNA sequence and truly quantify on a base by base level how different people are from each other, you're only kind of like estimating it. And I mean, to be honest, the ways they estimated it were pretty clever. There was another one, high resolution melting, which used the tendency of DNA to hybridize and looked at how similar individuals' DNA was based on how well it would hybridize between individuals. So there was a lot of stuff you could do, but the only way to really know for sure is to do a lot of DNA sequencing. And so this is what we know now thanks to the genomics age. So there have been a lot of large-scale projects such as the 1000 Genomes Project and the International HapMap Project, as well as the Genotype Tissue Expression Project, all of which have basically had the aim of characterizing the scope and the universe of human genetic variation in a wide range of populations. So most of what we're doing here, and most of what you'll learn about in this class, is kind of based off of those blueprints or those parameters established by these projects, and saying, okay, of the universe of variation that exists among humans, are there any variants that seem to be more commonly, that you find more commonly in individuals, say, with autism or schizophrenia? You're taking known variants and you're looking to see if they happen more often in people who are cases versus people who are controls, or whatever the model is that you're looking at. So typically not trying to find de novo mutations, although that does go on, and we'll certainly learn about that too. The typical way that the genetics of mental health are studied and the genetics of most common diseases are by trying to link normal human variation to the conditions and see if certain variants cluster with those conditions. Now, the kinds of mutations that can lead to this variation are substitutions. So this is just a basic kind of run through of the list. You've got substitution. This is when one base is swapped for another. This is the simplest and most common type. So single nucleotide polymorphisms, or SNPs as we call them, are this. An A could get switched to a T, or a C could get switched to a G, or a C could get switched to a T, that type of thing. And of course, where that happens really matters. You could have insertions. This is when you aren't changing a base, but you're actually adding a new base in. So you might have ATT, and then the mutated version could be ATCT. You've added a C in there, and you've changed the whole kind of framing of that sequence. There's also deletions. This is sort of the reverse, where you've taken away a base, and again, you've changed the way the DNA or the way the sequence is framed. There's translocations. These are not terribly common, but they are quite relevant to the process of evolution. This is when large chunks of chromosomes can be moved to different places and rearranged in a kind of structural way. There's also inversions. It's possible for the sequence of DNA in a given area to, through this mutation, essentially flip and reverse. So a gene is suddenly coded backwards from the way it used to be coded. That is possible. And of course, duplications. Genes or pieces of genes or other types of things, transposons and whatnot, can be duplicated and stuck back into the genome somewhere. And so a lot of actual relevant evolution takes place on the basis of this. If you look at how many genes for different, say, G protein coupled receptors are similar to each other, the reason is that there's sort of an ancestral G protein coupled receptor that would be duplicated. And then on the new duplicate version, you have kind of a starting point to add changes that won't impact the survival, because you still have a good copy of that gene that did its original thing. You duplicate it. You can almost, it's almost like, if you're working on, paper, and you save as and you make a new copy of it, you can work on the new copy and try out new stuff without ruining the original copy. This is sort of the essence of duplications. And so as far as evolution goes, that's quite useful. You wouldn't necessarily want all different types of your genome being duplicated, left and right without any kind of larger scale evolution. I mean, obviously, like the trisomies, for example, trisomy 21, Down syndrome, is a type of duplication. You're adding an extra copy of the 21st chromosome. So going to the chat here. So Ali asks, what are de novo mutations again? Right. So de novo means new, and de novo just means like a new mutation. So it's a mutation that you don't see in either of your parents. And that means that they didn't pass it on to you. It's a mutation you have because it was created sometime after your parents were, at some point after your parents' DNA was established. So it could happen in the mom's egg, or it could happen in the father's sperm. Mutations that happen there don't affect either of your parents because they're happening in the gametes, not their somatic cells, but they will affect you because, of course, you come from the gametes. So any mutation that exists in the child that's not present in either of the parents is taken to be a de novo mutation. So all of these mutations tend to take place during DNA replication. Not actually, I should say, like, this isn't like universally true. Transposons and whatnot can go to work in interphase. So there are definitely changes that happen to the DNA that don't take place strictly during replication, but this is when a lot of it goes on. And of course, if they occur in somatic cells, they're not heritable because you don't, if you have a mutation in a neuron, that's not going to get passed on to your offspring. But if they happen in germ cells, they are called germline mutations, and they will be passed on. And a germline mutation is typically also a de novo mutation because it's a new thing that appears in one of the gametes or one of the germ cells, and they'll be passed on. This is also like the source of a lot of people's worries about genome or gene editing. Because if you edit genes in your own like muscle cells, for example, if you edit something to make you have better or bigger muscles, that's not going to really affect your children or the human gene pool. But if you manipulate the DNA in your sperm cells, for example, that will be passed on forever and it'll never go away. As long as your offspring keep reproducing, that that gene editing will exist forever. So that's a bigger concern when it comes to ethics than something you do personally to yourself. So in the genomics age, which is the age that we're in, variants are often defined relative to the reference genome. Now, I said that there's no standard human being that you can compare everything to. That's true. But there is a reference genome that exists, and it's just basically the current one that we're using is HG38. This is the one I'm asking you to use in the assignment. This is just a version of the genome, a version of the reference genome. And it was assembled from 13 anonymous donors in Buffalo, New York. So they essentially just got 13 random people, sequenced their DNA to a very high depth, put it together and said, okay, this is the baseline genome, these guys from Buffalo. Now, that doesn't mean that this is normal or that this is the standard way that the human genome should be. All it means is that you've got to have something that's the baseline in order to make comparisons to. So, naturally, that happens in the place that's doing the most sequencing and has the most resources to do this. So this happens to be some random guys from Buffalo, but it's not that's the normal DNA. It's only the reference DNA. So there are other I think now, the upcoming versions of the human genome assemblies are being made in such a way as to have more different types of reference. It's very, it's kind of questionable how meaningful it is to like look at DNA, say from people in Nigeria and compare it to some 13 guys from Buffalo. Like, it's hard to say that that's going to be a great, all it'll give you, it'll tell you that people in Africa are genetically rather different from people in Buffalo, New York, but that's kind of trivial. What you really want to know is how people with the disease differ from control people in their own population. But anyway, it's not normal, but it exists, and this is sort of what we have to use. The reference genome is actually printed out in the Wellcome Reference Center, so you can go and just look at pages and pages of A, T, C, and G printed out on paper if you ever want to like feel the physical volume of the human genome. Anyway, so it's even though we're doing a lot more sequencing now, and whenever you want to study the genetics of this or that, you can do DNA sequencing, and you can have a very complete overview of your subject's DNA. It's still difficult to assemble brand new genomes. I guess ideally, every person you study should have a completely unique genome, and you'd want to assemble their genome from scratch. That's not really practical at the moment. Typically what happens is we use the reference genome to map our data onto. So when you're doing DNA sequencing, and of course we're going to have a lecture on that coming up soon, but when you're doing your DNA sequencing, you get little fragments of DNA that come through, and you have to, it'll be a little stretch of DNA from some completely random part of the genome. You get billions and billions of these little fragments of DNA, and you have to map them on to where they belong in the genome. And then once you've done that, you can look at any variants that might appear in those fragments. So you have to figure out where those fragments belong, and then identify what SNPs or what variants exist within those fragments. So you do need like a reference map or like an atlas to map these onto. That's what the reference genome is for. You do a bunch of sequencing or a bunch of genotyping, and then you use the reference genome as a template for where those data belong and how to sort of arrange them. And then naturally you end up using it as a bit of a comparison tool as well. But really the bigger purpose is just figuring out where all the pieces fit together. It's a bit like if you're doing like a jigsaw puzzle and you're looking at the box to tell you what the end result is supposed to be like. It's much simpler to do a puzzle if you have a picture of what it's supposed to be. If you're trying to figure out what the puzzle is supposed to be just by looking at the individual pieces, that's basically impossible to do. It is actually possible, but it's like incredibly labor intensive. So that's the kind of the idea of the reference genome, is it gives you a starting point, so you have to do less work to gather data and apply it. So we have another question. Ken asks, why did they only use 13 subjects? I'm not sure why they picked exactly 13. The reason they don't use very many for these is because they have to sequence them to a very high depth. So, and we'll talk about sequencing. I believe that actually might be the next lecture. But anyway, the basic idea is that in order to put the genome together, you have to have lots of different pieces to work with. It's a bit like if somebody took a book from the library, say like, whatever, A Tale of Two Cities or some kind of book, and they tore it up so that all the pages were just completely torn up, and you had to put them all back together. So you would look for sentences where, a sentence ends with, he went to the, and then it ends. And then you have to find a place that could fit with that. So maybe you would find store. Sorry. And you're trying to put it together. So it's quite expensive to sequence to enough depth to make this possible. And the trade-off is always between you can sequence a whole bunch of people at low depth, and then you're getting a kind of overview that tells you the big differences between people, but you wouldn't have enough data to assemble a genome. Or you can take a handful of people and sequence them to a very, very high depth and do all of the work needed to take those pieces and stitch them together into a genome. So it's kind of a trade-off between how much detail do you want versus how many people you want to look at. And so when you're doing a reference genome, you want a very, very high level of detail, and the trade-off there is you can't look at too many people because it just becomes too much to work with. So de novo assembly, here again, this is building a genome from scratch, a new genome, involves generating enough sequence data so that you can completely stitch the genome together. It's like I say, you've torn up a book and you're trying to put the pages back together. You need kind of, you almost need more than one copy of the book if the book is torn up completely randomly. You might have a sentence like, he went to the store, that's what it's supposed to be. And then on one fragment you went, he went, and then you have another fragment that says, to the store, and then a third fragment that said, went to the store. And when you put those three together, you can kind of stack them up and say, okay, these all fit together. This forms a complete sentence. It takes 2 books worth of pieces to do this because of where they're torn up. Maybe that doesn't make any sense, but I'll try to explain this analogy a little bit better when we talk about this for real. The point basically is, as I said, it's a trade-off between detail and sample size. And here, when you're doing this, you want the maximum amount of detail, and so you suffer in terms of sample size. So there's a great deal of human genetic variation across the world. Obviously, the history of genomics began in the places that were rich and developed enough to study them. So the United States and Great Britain tended to be the earliest developers of genetics and genomics technology. So they were their populations ended up getting a lot of the earliest amount of sequencing and the earliest amount of data. But now we've been able to branch out and look at more and more groups of people across the world. So here is a figure from the 1000 Genomes Project showing, and I don't, of course, don't need to memorize this or anything like this, but it's essentially just showing the share of genetic variation across all the different populations studied. And so I don't, have all of these memorized. They're available on the 1000 Genomes Project page, but each of these 3-letter acronyms refers to a specific group of people. So GBR, for example, is Great Britain. CEU is like Central Europeans. JPT is like Japanese, and so on and so forth. So you can kind of go through, and they've got like a fairly representative tranche of humanity. And what these pie charts represent is the fraction of the variation that's either private to the population or to the continent, or shared across continents, or shared across all continents. So you can kind of see that, like, okay, so like these people here have a fairly small amount of variation that's unique to them, and a fairly large part of it is in gray, meaning that they have a lot of genetics in common with everybody else on the planet. Whereas other groups have a larger fraction of variation that's unique to them. So this is just sort of a way to kind of eyeball this. Another way to look at it is with this figure here. What we have, again, from the 1000 Genomes Project is the number of variants variant sites per genome in million on the y-axis. So 4 million, 5 million per genome. And then on the x-axis, we have all the different populations studied. Each individual point here represents an individual person who was sequenced. So some people are kind of outliers and have a ton of variation. Other people are kind of closer toward the middle. So and of course, these are variants relative to the reference genome. So relative to the reference genome, people from Western Europe, Great Britain, Finland, Central Europe, these kinds of people, they are not terribly different from each other in terms of the number of variants they have compared to the reference. These people are all fairly similar to the reference genome, of course, because the reference genome was made up of people of European descent for the most part. As you move further and further away to Africa and Asia, what you'll see is that people in those populations are much more different from the reference than people from Western Europe. And this is not surprising, I suppose, because it's very clear that they're coming from a different population and they have a different background and a different history. And naturally, they're going to have more genetic variation relative to the reference genome than the people who essentially made up the reference genome. Now, it's maybe obvious, but maybe not. If the reference genome had been made up of say, for example, Mende individuals in Sierra Leone, MSL, if those people had been the reference genome, then this whole plot would have been reversed. They'd be down here, and all of the British people would be up here, and the European people would be up here. So it really just depends on who you define as the reference. And, not surprisingly, all the people who did this decided that they should be the reference, so they made it so. All that being said, the average 2 humans are extremely similar to one another when you talk about genetics. Any 2 people on Earth, if you were to look at their DNA, 99.99% of their bases would be the same. So almost everything in their genome is the exact same. Humans and chimpanzees are about 98% identical, which is also fairly similar. In the video for the assignment, I show you the ways to look at conservation in the UCSC genome browser. So you can actually see on a base by base or on a region by region level how conserved a given piece of of the human genome is, like how much of that is identical to, say, a rhesus monkey or to a mouse or a rat or something like that. So, the further away you move from humans in evolutionary history, the less and less similar the genomes end up being. But even still, humans and chimpanzees, fairly similar. So it's worth kind of bearing in mind that human genetic variation doesn't affect any of our most fundamental qualities. The things that really make us human, our intelligence, our emotions, our empathy, the way we love our families, and the way we react to trauma, this stuff is essentially the exact same among everybody. There's really no meaningful difference in almost anything. The variants we do see and really emphasize are actually quite superficial. If you think about the amount of emphasis that's placed on, say, skin color, relative to how important this is, like scientifically, it's kind of it's a bit of an indictment on human society, I suppose, because many of these outside phenotypic variants are meaningless or at least arbitrary from a biological perspective. Why would why would you prioritize one variant over another when it doesn't have anything to do with disease or with anything meaningful? So some of these are arbitrary, but of course they're visible, so we tend to focus on them. And of course, they also correlate with your culture and where you come from and so forth. So there's a lot of things that are tied up with that. But on a purely, on the level of pure molecular biology, these things are rather arbitrary. And then most genetic variation between populations isn't due to selection exactly. It's mostly just due to random genetic drift. If you take a bunch of people and put them on one continent, and then you take another bunch of people and put them on a different continent, and keep them separate for 50,000 years, they will end up genetically rather different from each other. Not necessarily because the environment has been selecting certain characteristics. It might just be because of random drift, and they're all kind of interbreeding, and there's sort of like an island effect there. So a lot of this doesn't even have to do so much with natural selection or survival of the fittest or any of that kind of stuff. It's just basically random drift and random change over time. And of course, with assortative mating, you're more likely to mate with people who are similar to you. So these random changes tend to become amplified over time because you possess certain random changes that make you look a little bit different, or I guess to be more accurate, they make you look a little bit more similar to everybody else, and you're going to try to favor people who are also more similar to you. So it tends to amplify this tendency toward drift over time. Now, the vast majority of mutations don't have any effect on phenotype, but they do still serve to distinguish individuals. So this is another important point, is that most of the human genetic variation and also many of the mutations we experience, you would never even know it was there because it has no visible effect on phenotype. Like I say, the things that we do notice, things like skin color and hair color and eye color and that type of stuff, that comprises a very small fraction of the real genetic variation between people. You have tons of variant between you and others that are totally invisible to you because they have no they have a microscopic effect on your phenotype or perhaps no effect at all. So I just pasted in a table from one of my textbooks that kind of go over this. And I don't want to, belabor these points too much, of course, because this would hopefully be stuff that's somewhat familiar to you all. But it just goes over the basic ways of classifying mutations. There can be origin. They are either spontaneous or induced, meaning that either they were caused by something like UV light or a mutagen, or they just happened by random chance. There's the cell type, whether they're somatic or germline. We talked about that. There's expression, whether they're universally expressed or if they're dependent on something. The effect on function, of course, can do lots of things. Molecular change, we talked about this, whether it's substitutions or transitions or transversions or insertions or deletions. This kind of transversion thing, I don't think that's really too relevant to us. That's kind of biochemically meaningful. It's easier to change one purine for another purine than it is to change a purine for a pyrimidine, but that's not something to worry about for us. And then, of course, effect on translation. Does it change protein coding or does it not? And how can it do this? So this is just kind of to review. It's not to, like I say, for the specific variants and mutations that we look at in class, we'll we'll study this in more depth. So let's look at mutations and variations in practice. Now, we could spend a lot of time trying to learn about how mutations happen and where variants come from. I think that's not going to be our goal today. And like I said, that's not necessarily even the goal for the whole class. For most of what we're doing, I think you can take for granted that the mutations and the variations exist. They come from somewhere. And what matters really is how they affect your phenotype, how they affect the kind of three-dimensional architecture of the genome, epigenetics, all this kind of stuff. Where they come from is almost kind of immaterial to for our purposes, unless I say otherwise. Most mutations, and in fact most variations, have no apparent effect on phenotype, and because of this, they're not selected for in any way that we can tell. They pass effortlessly on to the next generation. They clump up and they accumulate in places because of genetic drift and assortative mating, but they don't necessarily have a strong enough effect on your likelihood of reproducing that they would ever be subject to any kind of meaningful selection. In the genomics age, the traditional sort of Mendelian concepts of allele effect have receded in importance. So you might have, I guess, supposed that this is a genetics course. We should be hearing all about dominant and recessive alleles and this kind of thing. And that doesn't end up being a big part of what we do in genomics. The reason being, most of the conditions we look at are highly polygenic and involve hundreds, if not thousands, of different locations in the genome, all of which together add up to giving you a higher risk of a certain condition. And when you're talking about such a contingent, conditional relationship between genes and the phenotype, there's no way to really meaningfully talk about dominant or recessive things. That's just not the inheritance pattern that you see for most of the mental health conditions that we're going to be learning about. Now, I kind of thought of this little didactic exercise. You ever see these like iceberg memes where it's like, the top level is like, the stuff that everyone knows about, and then as you get deeper into the iceberg, you become more and more conspiratorial. So down here is like, oh, the reptilians control the government or something like that, and up here is just central banks are good, whatever the conspiracy theory you favor. But anyway, so I thought it's kind of a neat exercise, or like a neat kind of visual aid, I guess, to a point that I want to make about the way most variation plays out, and about the kind of the way that we think about these things in the modern context. So here on the tip of the iceberg, we're talking about genetic variation that influence very visible traits, things like skin color, hair color, other eye color, that type of stuff. These are typically coding because they typically affect the protein coding sequence, or at least they very directly affect whether a protein is or isn't expressed or is functional or not. So they're typically coding. Now then you have the genetic basis of phenotypes that are not obviously visible, like blood type. Blood type is, of course, a genetic thing. It follows the normal rules of Mendelian inheritance, but you can't see it on the surface, so you can't technically use it as a way to discriminate against people, but it is there, and it's genetically fairly simple, just as hair color and skin color tend to be. And then one step below this are genetic variations that influence known biochemical pathways. Now, this is where we're getting closer to the kinds of things that people study when they're trying to figure out the genetics of mental health. So a lot of work has been done on, say, for example, monoamine signaling, or serotonin or dopamine and norepinephrine and these kinds of things, or melanocortin signaling, MC4R, the melanocortin 4 receptor, pro-opiomelanocortin, these kinds of things. A lot of neuroscientists have spent a lot of time figuring out how all these systems work, mapping out the anatomy of the mesolimbic dopamine system and the raphe nucleus for serotonin and all the rest of it. And through this, they've built up kind of a concept of mental illness that is very centered in dysfunctions in monoamine signaling. Depression, of course, is treated quite often with antidepressants, which are serotonin reuptake inhibitors, or serotonin norepinephrine reuptake inhibitors. That might make you think that or that would make you perhaps believe that mutations to genes involved in serotonin signaling are responsible for depression. That's a very reasonable thing to do. So these are what I would call kind of a higher up in the like, you know, they're below the surface of the ocean, but they're still pretty superficial as far as the different kinds of relationships you can have between genetics and disease. And then hidden below this, you have the non-coding variants, of which vastly outnumber the coding variants. You know, most of, you know, your exons only make up about 1% of your genome, and variants to exons tend to be selected against. So coding variants that really influence the sequence of a protein in a deleterious way are extremely rare. However, variants that influence gene expression, and whether it's cell type or tissue-specific gene expression, those can be quite common because they don't have to happen in exons. They can happen in introns, they can happen in promoters and enhancers, they can happen in intergenic space, they can happen all over the place. And because they're not changing the actual coding sequence of a protein, they don't tend to be selected against to nearly the same degree. And then finally, the deepest level that I found were changes to chromatin conformation. That means the three-dimensional structure of the chromatin in the nucleus, as well as epigenetics and enhancers and so forth. So genetic variation as it relates to phenotype and disease could be at any one of these levels of the iceberg. You could have the very simple kind that appear at the very surface and just make you look different. You can have things that are below the surface that change hidden aspects of you, your metabolism, like whether you have PKU or whether you're AB positive or for your blood type or whatever else. And then you can have ones that affect pathways that are even more invisible than that, things happening in your brain, but that are still tied to existing knowledge about the brain and existing kind of neurotransmitter systems. Then below that, you can have mutations that don't even affect the coding sequence of genes, but change the way genes are expressed. And then below even that, you can have mutations that don't even affect the expression of genes, but just affect the epigenetics and the three-dimensional shape of the chromatin. So there's all different kinds of levels. And we're going to be spending most of our time, I think, down here in the deeper recesses of the ocean, because this is where most of the newest findings are. But I'm going to give you some examples of all the other ones as well, just so you know. So let's skip to variations in known biochemical pathways. One of the most common ways to look at the genetics of mental illness is by focusing on genes in well-understood biochemical pathways. As I mentioned, neuroscientists have understood how serotonin works for quite a while, and it's natural to assume that depression, being a condition that's treated with serotonergic drugs, must have something to do with mutations in serotonin. And I mean, it sort of does, but this is a hypothesis you can come up with without even looking at the genome. You can just think of this with just on a completely a priori basis because it depends only on what you know about neurotransmitters. And this is very favored by many neuroscientists because it fits very well into the existing scope of biology. It means all of the models they're using, all of the different perspectives they have on the condition are essentially correct and will tie in with the genetics very, very nicely. Another good thing about these types of studies is that typically a variance in known biochemical pathways can be easily genotyped because they're usually just one particular allele that changes a certain protein, a given enzyme or a receptor or something that processes or receives the neurotransmitter. So if you can genotype people easily at one locus, that makes it very easy to design experiments because you can just get a whole bunch of people, divide them into two groups. They either have the mutation or they don't. You know, they either have this allele or that allele, and you can do a whole bunch of tests on them and then just compare the two groups and see if there's a difference. Now, so I guess the one that maybe you have heard of or might be most familiar to you are variations or polymorphisms, I should say, in dopamine-associated genes. So these are all the genes that either act as receptors for dopamine or as transporters or as enzymes that play a role in either synthesizing or degrading dopamine. So enzymes like COMT, catecholomethyltransferase, DBH, dopamine beta hydroxylase, monoamine oxidase A and B, tyrosine hydroxylase, which is involved in synthesizing dopamine. You've got the different dopamine receptors and, of course, the transporters that move dopamine in and out of vesicles and so forth. So these are all proteins that are very well studied in mouse models and even to a certain extent in human beings. And, of course, there's lots of diseases we know about that are very heavily rooted in dopamine signaling. Think of schizophrenia, think of Parkinson's, think of, well, I'm just thinking of those two at the moment, but there's lots of other ones too. Huntington's, I suppose, as well. So the thinking would be then that maybe these conditions have something to do with mutations in one of these specific enzymes. And you've narrowed it down quite a lot by doing this, because instead of thinking about the 20,000 genes that exist in the human genome, now you're dealing with maybe a dozen or so, maybe, half dozen. You've narrowed it down considerably, and that makes your job as a scientist much, much easier. So you can see why this type of study is rather popular. So the classic one that you probably have heard of is the classic Compt-Val-Met allele. So this is a polymorphism in the catechol-O-methyltransferase gene. This is a gene that's involved in breaking down dopamine by adding a methyl group to it, and it goes through sort of a degradation process. And it involves a swap from at the 158th amino acid position in the protein's sequence from valine to methionine. This is a coding SNP, so it takes place inside of an exon, and it's simply a SNP that changes an A to a G, and this affects the 158th amino acid, and this protein, or this enzyme, breaks down dopamine and other catecholamines. So you change the actual protein that's doing this job. And because this has been so well studied, it's been in all the different books and everything that everyone's ever heard of, they've kind of come up with like a shorthand for it. The A, or the Met allele, is described as the worrier, while the G, or the valine allele, is described as the warrior. So it's the worrier versus warrior genome. And the Val allele, the warrior, produces an enzyme that has higher levels of activity than the other allele. So that means dopamine is broken down faster in people with this so-called warrior allele, and people who are homozygous for it, i.e. they have valine, GG alleles, they are the ones with the absolute highest rate of dopamine breakdown. Now, you can look at the allele frequency for this, and so this is data from the International HapMap project. You can actually find this on SNPpedia if you look up different SNPs that you're curious about. This one's RS4680, so I believe you could, if you wanted to, you could even look this up on the UCSC genome browser following the directions that I give and find out where that's located and learn all kinds of fun facts about it. But anyway, this gives the allele frequency, so out of, it goes up to 100, of course. And you just look at the different frequencies. So among Europeans, the GG genotype, who are homozygous for the Val allele, makes up about 25% of people. People who are heterozygous make up about, people who are homozygous for the A allele, or for the worrier allele, in Europeans make up about, let's say, a little less than 20%. So you've got about, or a little less than 25%, I suppose. So you've got about maybe around half the people are homozygous for either of the alleles, and the remaining half are heterozygous. But this frequency varies across populations. So you have a higher proportion of so-called warriors among the Han Chinese, CHB is Han Chinese. So among, it's hard to see here. Han Chinese, you have a higher proportion of so-called warriors than you do among white European, or whites of European descent. So the frequency of this varies across people. And so I guess maybe you can look at this as like a microscopic illustration of what allele frequency means more broadly. The different versions of this gene are not equally distributed among the world's population. Some people have more of one versus the other. So the Val allele results in lower dopamine tone in the cortex, of course, because it's metabolizing dopamine more rapidly, and thus there's less of it floating around in the cortex to stimulate the receptors. You can look at PET imaging actually with radio-labeled D1 ligands, dopamine receptor 1 ligands, and see that it binds differently in people with this Val allele compared to people without it. It actually, it shows higher levels of ligand binding because there's less natural dopamine for it to compete with. So what you actually see is higher levels of D1 binding in Val-Val and Val-Met people than Met-Met people. So there might be upregulation of the receptors to compensate for the fact that there's less natural dopamine around, or it could be the fact that there's just less dopamine for this ligand to compete with. In either case, the presence or absence of this warrior Val allele determines the amount of dopamine that's available for your prefrontal cortex. And according to the theory, the less dopamine you've got floating around in your prefrontal cortex, you have a lot of different behavioral and personality changes. So a couple of studies, and you can, like, if you go to the databases, you can see just countless studies that have been done using this particular genotype. But one of them that's rather famous came from Caspi et al. In 2005, where they showed that adolescent cannabis use can increase schizophrenia risk, but only in people who are homozygous for the VAL allele. People who are homozygous for the MET allele, this apparently doesn't happen. So this is an interaction between your risk of a certain condition and your genotype and the environment, because cannabis obviously is not part of your genes, it's the environment. So you've got a gene by environment interaction there when it comes to schizophrenia. The antidepressant paroxetine, or Paxil as it's often called, seems to work better in MET individuals for some reason. And also MET individuals have higher pain sensitivity, meaning that they're not warriors, right? You can't have a warrior who gets hurt really easily. So these fellows, or folks rather, who are the MET carriers, they have higher pain sensitivity. Now, on kind of a practical note, this is very easy to do these kinds of studies because you just have to genotype one locus. This is a SNP that happens in one place. To genotype it, you just have to have a PCR assay or something like that can be run very simply. You can study hundreds of people and do very elaborate psychological experiments with them that wouldn't be possible using the normal genome-wide study format, because genome-wide studies take tens of thousands of people as subjects. You can't do tens of thousands of psychological experiments. You have to have a smaller number of subjects. So it's easier to do these kinds of studies. Part of the reason is because if you're only looking at one thing, you don't have to worry about genome-wide significance. If you've taken stats class before, you know about testing correcting for multiple tests, like the Bonferroni correction and these kinds of things. That same logic applies to the genome. So if you're studying, say 100,000 different SNPs, you have to take your p-value and basically divide it by 100,000. So things have to be very, very significant if you're looking everywhere. But if you're only looking at one thing, p less than 0.05 is good enough. So it's much easier to do these kinds of studies for a number of basically practical reasons. That's why this has been so well studied, despite it being just one SNP out of zillions that human beings have. Now, the next level below that are non-coding variations that lead to changes in cell and tissue-specific gene expression. So this, by the way, is important to bring up. This is going to be quite relevant going forward. The genome is the same in every cell in your body. I can take blood cells, or I can take hair follicle cells, or I can take cells that I scrape off of the inside of your cheek, wherever. You're going to have the exact same DNA in all of those cells. Gene expression is not the same everywhere. Gene expression is very different depending on where you look. Your skin expresses different genes than your liver. Your liver expresses different genes than your neurons. Your neurons express different genes from your glia. So even within the same tissue, i.e. the brain, different cells can express different genes. So if you want to know what how a given genotype is affecting gene expression, you have to look exactly in the tissue that you think it's happening in. If I think that a given gene is influencing gene expression in the brain, I have to look in the brain, which is where the difficulty lies, because it's very tough to get brains out of people. You have to have post-mortem samples, and it's a whole big production. So, but I just want to I want to sort of stress that only the genome is universal between all cells. You have to study tissue, or you have to study gene expression and also epigenetics in the tissue that you're curious about. Anyhow, so the kinds of variants that affect proteins are either missense or nonsense. They can affect the actual coding. Missense SNPs are ones that result in a codon being switched for something else. A codon is just a three base sequence that denotes a certain amino acid, and you can switch one of the bases there and sometimes get a different amino acid that changes the way the protein is. You can also have nonsense mutations that change a codon into a stop codon that makes the protein shorter than it should be and typically ruins the protein's function. However, about 90% of SNPs that affect phenotype are located in non-coding regions. So how could a SNP that doesn't change the protein code or the structure of the protein possibly affect phenotype, right? This is the big question, because when you look at the genome-wide studies that we do, most of the changes happen in non-coding regions. They must therefore affect gene regulation, not protein structure. And as I said, gene expression is tissue and cell type specific, meaning you have to look exactly in the right tissue. You can't just look in some random tissue you happen to have access to. It has to be in the exact tissue you are curious about. Some variants, for example, can only affect gene expression in a particular part of the brain, not even just the brain as a whole, but some genes may only change something in the dentate gyrus, and you'll never know about it unless you actually look at the dentate gyrus. So the level of complexity becomes exponentially greater because you have to kind of look in every tissue and potentially in every cell type to even know. Luckily, we don't have to do that because there is a project that's already been done to try to figure out a lot of this stuff. It's another one of these big consortium deals where a bunch of scientists get together and produce a vast quantity of data to just use as a reference point for everybody else. Not everyone can afford to do these huge experiments, but everyone can benefit from having the data. So this is the Genotype Tissue Expression Project, GTEx. It's a large-scale consortium, and what it's trying to do is combine DNA sequencing and RNA sequencing to explore the effect of genetic variation on tissue-specific gene expression. So they have a bunch of cadavers that were donors of tissue, and they extracted RNA, messenger RNA to be precise, from all these different tissues. So look at all the different things they have. They've got different parts of the brain, anterior cingulate, they've got cerebellar hemisphere, they've got cortex, frontal cortex, hippocampus, hypothalamus, so on and so forth. They've got liver, stomach, adrenal gland, adipose tissue, blah, 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 like the whole works. They've got tissue from every part of the body, all three germ layers are represented, all the different main things anyone would ever care about. Skin that's sun exposed and skin that's not sun exposed, all that good stuff. And they've got expression data from all these things, all these tissues. They can measure gene expression, how much of a gene is expressed, as well as alternate splicing, whether certain exons were excluded or skipped over. That can be quite relevant for the function of the gene as well. And then because they've got the genotype data, they can look to see if there are genetic variants that affect the expression of a particular gene. Not the structure of that protein, but just the amount of the protein that's produced by changing the amount of mRNA that's produced. So variants that affect the level of gene expression are called EQTLs, or expression quantitative trait loci. You may have remembered QTLs, quantitative trait loci. These are regions of variation that correlate with quantitative traits like height or BMI. However, in this case, they're correlated with expression of a given gene. So here's an example of one of these that they found. Now, this is a rather complicated diagram, so I'll try to walk you through it. So starting from the top, this is a map of where this SNP is located on the chromosome. This is a single nucleotide polymorphism. You change one letter of the DNA, this is what you get. So it's located on here, the long arm of chromosome 11. It's located right here. It's called RS633185. You can look that up on SNPpedia or using the genome browser if you like. Now here's a close-up diagram showing all of the different genes that are located around where this SNP is. So you can see that this SNP is in the middle of the RGAP42 gene. The gene starts here and it points this way, so it's transcribed in this direction, and it's actually located inside of an intron. Now, of course, introns are not protein coding, so this SNP can't possibly affect the protein RGAP42, whatever protein that gene codes for, but it can affect the expression of it. So here's a very simple representation of what they find. There is this allele, I guess this is the 633185 allele, and you can either be homozygous for the reference allele, meaning you have whatever allele was arbitrarily chosen as the reference. You can be heterozygous, or you can be homozygous for the alternate allele. This is just the other possible allele. It's in this case, it's either a G or a C. So you could either be homozygous for the G, you could have a GC, you could have one of each, or you could have homozygous for the alt and have CC. So depending on which of these genotypes you have, the expression of this gene, TMEM133, is decreased. So if you're homozygous for the alternate allele, you've got lower expression than if you're homozygous for the reference allele. Note that TMEM133 is located right downstream. So it's actually the case that this variant actually influences the expression of TMEM133 Another nearby variant actually affects RGAP42 to the sort of the same way. So in both cases, you've got a variant that's nowhere near the gene that's actually being regulated. It's inside the intron of one gene, so it can affect that gene. But it's also nowhere near TMEM133, yet it's able to affect the expression of it. So that if you have GG, you're going to have higher expression of TMEM133 than if you have CC. And this SNP was already known by other people to affect blood pressure. Now, thanks to GTEx, we know that in the tibial artery, if you have the homozygous, if you're homozygous for the alternate allele, you'll have lower expression of the blood pressure regulating gene RGAP42 as well as TMEM133. So I guess this is kind of coming out to be a bit confusing, but the basic point is this. Here are genes, I should say, that are known to influence blood pressure, RGAP42 and TMEM133. The amount of the level of expression of these genes in the artery is influenced by what version of this SNP you have. It's really just that simple. The level of expression you have is influenced by what your genotype is and what SNP you have. It doesn't change the structure of either of these two proteins, but it does change the amount of it you have. And you only can see this if you look in the vascular smooth muscle tissue. If you were to look at this relationship in the brain, it wouldn't be there. If you were to look at this relationship in the muscle, it wouldn't be there. Blood pressure is largely a vascular thing, so in order to understand, you have to look in the vascular system. That is what I'm getting at here. And it would be invisible to you if you didn't do this. So thank you to GTEx for going through this and doing all of this work for us. Now we can use this database and look at where genes are expressed and how they vary depending on your genotype. You can also have epigenetic changes. So sometimes you may not even notice the effect of a variant on steady state gene expression. You might notice it on gene regulation, perhaps activity dependent regulation. Maybe you've got a variant that doesn't change the amount of a gene that's expressed at baseline, but it does change how much that gene is upregulated, say by stress. Then you have something that you wouldn't even see if you did the GTEx method. You'd have to do a very specific experiment to see what's happening there. You could also look at epigenetics because epigenetics influence how genes are regulated. So DNA methylation, as we will learn, regulates cell type specific gene expression potential, and genetic variants can influence DNA methylation. They can also influence microRNAs and histone modifications and all kinds of other things that we're going to learn about later on. But these are very subtle. Sometimes they're not even evident as changes in expression. They're below that level even. And of course, they are also cell type and tissue specific. So here's an example of this. Here is a SNP, RS2108854. This is what you would call an MQTL. This is a variant that affects the level of methylation at a given position in the genome dose-dependently. So not only is this affecting methylation, not expression, it's affecting it in a age and tissue-specific way. So extremely complicated relationship. Here's a box plot where the y-axis is showing DNA methylation measured in percent from 40% methylated all the way up to 90%. Here on the x-axis of the box plot, you have the different alleles that they studied. Somebody could be homozygous for A at this particular SNP. They could be heterozygous, or they could be homozygous for G. And this is the same SNP studied in 4 different brain regions, okay? So in the fetal brain, the more copies of G you have, the higher your methylation is going to be. So AA has the lowest methylation, and GG has the highest, and AG is in between. Okay, so that's a dose-response relationship between the presence of this particular SNP and the level of methylation in the fetal brain. But guess what, folks? It doesn't exist in the prefrontal cortex of an adult, nor does it exist in the striatum. Here, they're all around 79 or high 70s, low 80s as far as methylation percentage. So this is a variant that affects epigenetics at one specific time point during development. That, of course, can cause all kinds of things that we don't even really know about. I mean, epigenetics do a lot, but you wouldn't even know it was there unless you looked at this exact tissue and this exact time point. If you looked at fetal, skin cells, you wouldn't see this, most likely. And then in the cerebellum of an adult, the relationship is actually the reverse. So the more copies of G you have, the less methylation you have in the cerebellum, which is even more complicated. So this isn't the kind of thing that you really want to, agonize over on an individual level. It's just one example of how genetic variants can influence epigenetics in these really complicated ways where you have to look at a specific tissue and a specific time point or a specific, or in response to a specific stimulus to even see what's going on. So this is way below the level of like hair color, right, which is blindingly obvious to everybody. These are changes in the genome that affect the very subtle aspects of biochemistry and molecular biology. And this is what most of the genetic variation we study actually is. Most of the variants we study don't affect the protein sequence and make you have like more or less enzymatic activity. They affect gene expression and epigenetics and very, very subtle things in a tissue, cell type, and stimulus/time sensitive way. So just try to take that all in, I guess, and try to reflect on that as evidence of how complicated things can get. We'll see this. This is a taste of what's to come. The effect of genetic variation goes well beyond this. We're going to be focusing mostly in this kind of realm, the stuff that's below the surface of the ocean. All the surface stuff is all well trodden. Where the action is down here in the deeper depths of the ocean. So we're going to try to show you how these things are discovered, how they're studied, and what we can do about it. So I'll pause briefly for any questions that might have cropped up. I have a number of slides to go still, so I don't want to get too bogged down. But if anyone thinks of a question as we go forward, just feel free to type it into the chat, and I'll come to it when I get a chance. So now we're going to switch gears into behavioral genetics. And this is a part that's kind of a cousin, I suppose, to what we're studying. It's a field of study that doesn't really use our type of methods, and it doesn't really answer the questions we're most curious about, but it is an ancestor and a current day cousin of what we do. People have thought that personality and behavioral characteristics relate to genetics for quite a long time. Basically, as long as selective breeding has been around and done in a rigorous way, people have realized that behavioral and personality traits are heritable. The modern form of it, of course, came around in the 19th century, created by Sir Francis Galton. He was Darwin's brother, and in the 19th century wrote the book Hereditary Genius. And you can almost imagine how bad that book must have been when you look at this guy and you just think of the name of a book, Hereditary Genius, and what somebody in the 19th century would think about that. He essentially argued, or discovered, quote unquote, that eminent individuals, people who were very highly placed in society, were more likely to have eminent individuals in their close family. So, lo and behold, if your dad owns a dealership, it's very likely that you're going to own a dealership too, or at least be a successful person in some kind of respect. It's the most basic fact we know about how wealth and power tend to be inherited. It has really nothing to do with genetics, but this was the 19th century, and people really loved the idea that you could make racism scientific, because people were already sort of racist, right? You don't need science to be racist, but if you could add science in, that would make you feel even better about being racist. So that's kind of the essence of Galton's work, and it's really not good stuff. I mean, he also invented the field of eugenics, which is literally the inspiration for the Nazis, the idea that the human gene pool could be perfected by eliminating undesirable people from the breeding pool, led to untold amounts of horror. And I think it's worth bearing this in mind, because we're here learning about genetics as well, and there is some responsibility to not repeat these past mistakes. Now, I know if you go on YouTube long enough, you're going to meet all kinds of people who are bringing out the calipers and measuring people's skull sizes and so forth, talking about the genetic incompatibility of different races and so forth. This is kind of almost had a modern day revival. But if you want to know the science of it and you want to study this on a serious level, you have to be aware of what this can lead to and what people used to think. These ideas are very attractive because they justify existing patterns of thought, but they're not good. So virtually all psychological traits show some degree of genetic influence. These are mostly determined by twin studies. So looking at twin studies of intelligence, personality, schizophrenia, autism, hyperactivity, all kinds of things, they show a greater level of correlation in identical monozygotic twins compared to dizygotic twins. And the overall purpose of twin studies is to estimate the proportion of variability that can be attributed to genetics. So for example, general intelligence, the heritability is supposedly around 50%. This is often contrasted, and we're going to learn about this. The heritability estimated by twin studies is quite high typically. Intelligence is 50%. If you look at the actual genetics using genome-wide methods, the kind that we're going to focus on, the actual amount that the genetics we know about that can explain is maybe closer to 4 or 5%. So the heritability of intelligence is either really, really high, or there's a big mistake in how these studies are done. But in any case, it's quite controversial, and this is called the missing heritability paradox. The fact that heritability estimates from twin studies are often quite high, implying that genetics makes a big difference in things. Then when you actually look at the DNA using our best available methods, the amount explained by genetic variation is extremely low by comparison. We're talking a difference of like 50% down to like 5%. So there's this huge amount of missing heritability that we think exists, or people believe exists, that can't be identified using the normal, using the best methods we have available. Now that doesn't mean it's not there, it just means we can't find it. So twin studies take advantage of the fact that the average monozygotic or identical twins share 100% of their genes, and the average dizygotic twins share about 50%. So you can take measurements of twins, both monozygotic and dizygotic, and partition the variation in a given trait, say intelligence or educational attainment or reading ability or whatever else, into 3 of the following categories. There's genetic / heritability, which is A. This is how it's denoted in the classic model. There's shared environment, things that they have in common, C. And then there's unshared environment, things that are not in common between the two, things, E. So of course you've got your genes. If you're monozygotic, they're 100% identical. If you're dizygotic, they're 50% identical. You've got your shared environment. Monozygotic twins and dizygotic twins typically grow up in the same house and go to the same schools. And so a lot of the aspects of their environment are shared. But then some of them aren't shared. Maybe one of the twins joins the swim team, the other twin joins the chess club, that kind of thing. So all of the different determinants of behavior or phenotype theoretically can be broken up into these 3 categories. And when you add together all the different contributions they make, it should add up to 100%. The basic datum used is the correlation in a given measurement, a trait, say intelligence or whatever, between twin pairs. And you just look at the difference in correlation between monozygotic and dizygotic twins, and that gets you started. So this is the actual math of it. I don't know if we really need to emphasize this a huge amount. The basic idea is this. You can take the amount of correlation between monozygotic twins and the amount that's dizygotic, and by rearranging the equation, you can figure out the contribution that's made up of genetics, because you know that dizygotic twins have half the amount of similarity in their DNA than monozygotic twins. And then you can do some other algebraic stuff, and ultimately what you will get is a combination of the factors A, C, and E, all giving you a certain fraction of the overall variance explained by the different components. So if twins, for example, are identical, then 100% is A, and the other parts make up 0. This would be something like skin color. It doesn't really matter where you grow up. If you have the genes for, say, having like white skin, it doesn't matter really what happens in your life. That's what your skin color is going to be. So it's 100% genetic. Things like height are not, because you might have genes to make you tall, but if you're in an environment with poor nutrition, you probably won't grow as tall as you could be if you had an identical twin in some other country that had more food. So I'll leave it there. I think it's probably just not worth getting into in a great deal of detail, because we don't really talk about twin studies too often. But I did go to the trouble of working out an example for you by using a made-up tool. So please take a look at this slide on your own time and try to read through it and just get a sense of what's happening here, the way variance can be partitioned from these different causes. So one principle of behavioral genetics that has turned out to be true is the fact that complex traits are caused by many genes of a small effect. So back in 1978, DeFries and company were selectively breeding mice for open field activity. And the way this would work is you would just get a whole bunch of mice, you would measure how much they run around if you just put them in an empty box, and then you would divide them up into high runners and low runners. And what you would do is you would just take all of the low runners and breed them together, and you would take all the high runners and breed them together. Then when they had offspring, you would do the exact same thing on the offspring. You would measure the running of all of them, take the highest out of the high runners, breed them together, take the lowest out of the low runners and breed them together, and so on and so forth. And you would also breed a third set of mice without doing any type of selection. You would just breed them on a completely random basis. And what they found is that over the generations, they did like 30 different generations of this. It would have taken years to do. A study like this would never have been funded today, because today funding is not done on this kind of long-term basis. But what they found is that the open field activity of mice bred for high activity continued to go up. Even after 30 generations of breeding, it was still going up, while at the same time, the field activity of low activity mice was continually going down. Now, this leads to kind of an important inference, because if there was just one gene that controlled for all of the difference in activity, you would expect that after a couple of generations, every mouse would have this gene or have this allele, and then the difference would plateau. And the same, of course, would be happening on the low side. It would plateau, and you would never really see a change after that particular variant had reached fixation in the population, if it's just one variant. If it's a lot of different variants, each of them adding a small piece to the puzzle, then it takes much, much longer for them to all reach fixation in the population. Because, you might get some mice that have this, this, and this, but they're missing that and the other thing. And it just takes a long time for them to all accumulate. So that would be consistent with the idea that even after 30 generations, the performance is still changing. It hasn't plateaued. You haven't found all the genes and isolated them into one population to create a high level of homozygosity for those genes. So that's a fairly simple illustration of the principle we observe from genome-wide studies, which is that most traits involve thousands or hundreds of thousands or hundreds or thousands of variants, all of which have a very small effect. This is what's going on in this running study. You could probably keep doing this for another 30 generations and the running would just keep diverging. This is because lots of tiny different variants are all contributing to it, and it takes a long time for these all to accumulate in one population and reach fixation. Another principle that's unrelated to the last one, but it's also true, is that the heritability of intelligence increases with age. So this is measured by twin studies, and it shows that if you try to estimate what proportion of intelligence comes from genetics, the amount that you get if you measure people in childhood is lower than if you measure them in either adolescence or young adulthood. So if you were to measure children, you might think that intelligence is about 40% heritable, but if you were to measure teenagers, young adults, I guess, age 17 to 18, you'd come up with a number closer to 60%. So this is rather strange, because genetics are genetics. They don't change once you're conceived, essentially. So how could the heritability increase with age? This happens fairly often, and in this case, it happens at the same time as the component of shared environment decreases. So as kids age, they begin to have less and less in common with how they share their environment and how they spend their time. And at the same time, the genetics of their intelligence begin to have a bigger and bigger effect in the measured intelligence. And I guess the thinking would be that whatever genes you have that play a role in how intelligent you are not fully able to be realized early on, because I guess there's just not enough to test on, or there's not enough variation in the environment. You're all in kindergarten. How smart could a kindergartner be? That kind of thing. But anyway, it's a rather interesting puzzle that shows even something as simple as how heritable is intelligence is not not a yes or no question. It depends on when you measure it. Obviously, it depends on how you measure it. But it's a rather complicated question because it's not stable over time. Now, another point that's rather important to make is that environmental factors interact with genetics. So consider the fact that this is a hypothetical example given by these authors cited here, but even traits that are completely heritable do depend somewhat on the environment. Take, for example, this example they give for sunscreen. So imagine there is a gene that changes your risk for skin cancer, right? That's what's being plotted here. So if you have the gene, you have a high skin cancer risk. And if you don't have this allele, you have a medium to low skin cancer risk. Now, imagine what having or not having that allele would mean in either Rio de Janeiro in Brazil or London, UK. Well, folks, there's no sun in London. It doesn't matter how many alleles you have, you'll see the sun maybe 4 days out of the year. So even if you have the world's worst genotype for skin cancer, the odds of getting skin cancer if you live in London are lower than if you live in Rio. And if you add sunscreen to the mix, which you can choose to use or not, it'll affect that even more. So if you have the world's worst genotype for skin cancer, but you live in London and you wear sunscreen every day, you're going to be very well protected, and the effect of that genotype will seem way less than if you have that genotype and you live in Rio and don't believe in sunscreen. And so you can see this applies everywhere. The genetics you have very often depend on the environment that you're placed in and what you do or do not do or what you have access to. This is why I think, whatever you could say about the genetics of intelligence, basically not something you'd want to worry about as long as social inequality exists, because you can never really dissociate the effect of upbringing and living conditions from so many of these relevant things. So the point is that the environment interacts with your genetics in many, many complicated ways. They can both mitigate and exacerbate trait and disease-associated genetic effects. You also have cultural and genetic influences interacting. So there's many cases of this. There's one, for example, by Samuelson et al. Back in 2008. And what they found was that the heritability estimates for reading and spelling in school children, very young kids, differed substantially between Australia, the US, and Scandinavia. Now, we're all human beings, and in this case, the US, Australia, and Scandinavia are all essentially human beings, at least the people studied, were of white European descent. So genetically fairly similar. How could the heritability estimate for reading ability and spelling differ so much between these different countries? Well, the answer is that the education systems are different in these countries. So Australian kids begin learning how to read when they're in kindergarten, while in Scandinavia, kindergarten emphasizes social, emotional, and aesthetic development. I have no idea what aesthetic development is supposed to mean, but they're not really trying to teach kids how to read at that young age. So how on earth could you test whether reading ability is heritable in young kids if some kids haven't even learned how to read yet, or haven't learned as much about how to read? That all washes out, I should say. By the time you look at when they're older, the difference disappears. But when you look at a younger age, the effect of the environment is much stronger than the effect of the genetics. And this is just one of many such cases where a gene's influence on a trait can only be realized in the appropriate environment. Stephen Jay Gould actually had I think it was Stephen Jay Gould, who was an old paleontologist / biologist. A lot of great books coming from that guy. Sadly died a number of years ago of cancer. But anyway, he had a great quote that said, I somehow care less about the convolutions and characteristics of Einstein's brain than the fact that there were probably, I'm butchering this quote, but he cares less about the details of Einstein's brain than about the fact that there were probably hundreds of Einsteins toiling in rice fields and in farms somewhere, never getting a chance to experience their brilliance. So the idea being that you could be the world's smartest guy, you could be Einstein or Isaac Newton or whoever, but if you're born in a place where you can't use your brain because of your living conditions, it's irrelevant. You'll never realize your full potential. So whatever you want to say about heritability, it depends a great deal on the environment for it to be realized. And so my last slide here is just a nice illustration from this paper. You can look up these authors and find this paper. It's a good read, actually. But essentially, they're just talking about how complicated it is to predict heritability of things and how genetically influenced a trait is, because there is this kernel of actual genetic heritability that is true, but whether you can measure it and how big an effect it has depends so much on environment and culture. So of course, many of these things come down to wealth. You have the amount of wealth you have in your society determines how your home life is going to be. It's going to determine if you have lead in your drinking water, what kind of schools you can go to, whether you have Wi-Fi at home. It also affects how clean things are, whether you're exposed to diseases or parasites, and what medicines you have, and all the rest of it. Prenatal environment, everything essentially is kind of downstream of wealth. That's a sad realization, but I think it should be obvious if you've been looking at the major complaints we've had with society lately. It really comes down to wealth distribution more than anything else. We're more than rich enough to give everyone everything they need. It's just how the money or the wealth is distributed. But this all is sort of superimposed on genetic variation. These things are all linked. Disease resistance, nutritional uptake, executive function, memory, attention. These depend on your schooling and what type of resources you have. Your disease resistance depends not only on what you're exposed to in terms of diseases, but also what you've been nourished with. The whole thing is all very interwoven and very complicated. So I'll give you this as kind of a disclaimer, I suppose, before we go on, to say that while we are studying the genetics of disease and mental health, and we're doing it in a way that is as scientific as possibly can be done in today's era, you still have to bear in mind that this is what's going on in the background. All these variations in very changeable, very mutable environmental characteristics influence how well these genes are expressed. They might conceal heritability in certain cases. They might exaggerate it in other cases. So just keep this in mind. We're not able to do, we're not really here to do a class on like the effect of lead in drinking water, but there are classes about that, developmental toxicology and so forth. Just keep in mind this is all going on in the background. So we're focusing on the genetics, but this is all part of a much bigger picture involving all of the different layers of justice and inequality in society that all bear on genetic variation and so forth. So I'm going to leave it there. I'll just hop over to the chat. I did get a question during my little rant here. Nathalie asks, would we be responsible for reading a map similar to the expression, the EQTL slide? I'm assuming for an exam. That is potentially true. Let me zip back to that slide here. So I don't want to, where is it? Okay, yeah, so here. I wouldn't give you this. This is too much to deal with on an exam. You will, I think I might ask you to look at things like this, these kinds of genome browser images and figures like box plots and stuff like that. It is possible I could ask you for that type of thing. Certainly understanding these maps of the genome where you see different genes laid out on top of each other, pointing different directions with different kinds of features in them. That kind of thing is going to be quite important. That's sort of the main lesson I want you to learn from the first assignment where you're using the genome browser. You want to see how the genome is physically laid out and what all the different symbols mean. So, sorry, I was circling this, but I didn't have the slides up. Yeah, so this, I don't think I'd ask you to interpret this for an exam, but certain pieces of this you may, understanding how to read a box plot and what it means to say that there's a dose response curve between genotypes where if you're homozygous for the reference allele, you've got more expression than if you're homozygous for the alternate allele. That kind of thing could be in there, but I'm not going to try to torture you with some kind of crazy weird diagram that you've never seen before. It would be a very if I do it, it would be a very schematic version that should be pretty straightforward if you've been following along. All right. So any other questions from the audience before we sign off for today? I guess we're a minute over time now. We're a minute over time, but there's no bell. Nobody can stop me from continuing to talk. All right, then. Well, thanks to everyone for listening today. I hope this was interesting for you. Please let me know if you have any questions. We'll see you again on Wednesday and then also on Thursday for office hours. So, have a good day, folks, and enjoy the rest of your classes. We'll see you next time.